First, John, welcome to the show in studio. Thanks for being here.
Hey, thank you, Michael.
Tell us about your guest, and I think we've got Dr. Elliot Goldstein. Welcome to the show.
Thank you.
You're welcome. Please, John.
All right, yeah, let me get started. So today, as you mentioned, we got the CEO of ProMIS Neurosciences, Dr. Elliot Goldstein, on our show today, CEO Money. ProMIS's ticker, for all you listening, is PMN on the Toronto Exchange and ARFXF here in the U.S., and ProMIS is focused on finding a cure for Alzheimer's and other neurodegenerative diseases. The company launched in mid-2015 out of the University of British Columbia and has since been heavily involved in preclinical work preparing their lead compound for human trials next year. 2018 has been a rough year for Alzheimer's. Eli Lilly, Merck, Axovant, VTV Therapeutics, all announcing failed phase 3s. But as we'll find out today, all these studies were focused on the wrong thing. And with that preface, let me introduce Dr. Goldstein, CEO of ProMIS Neurosciences, and he's held senior positions at Sandoz, GSK, and other smaller biotechs, both in the U.S. and Europe. Dr. Goldstein, welcome to CEO Money. How are you doing today?
I'm fine, thank you, and pleasure to speak to your listeners today.
So how did you end up at ProMIS?
Well, I've been 35 plus years in drug development. That's my passion. In 2014, my business partner, Gene Williams, who's the executive chairman of the company, and I discovered, if you like, at a high-level scientific meeting, a presentation by the now chief scientific officer of ProMIS, Professor Neil Cashman. And to put it frankly, we were bowled over by the promise of this technology. So we were brought on, Gene Williams and I, as consultants to the company to help with strategic direction, given all of our experience in drug development, and we launched the company in July of 2015.
Great. And then can you give the audience just a quick explanation of what Alzheimer's disease is and how it causes memory loss?
Well, I'll try and be very brief because it's quite a complex disease.
Sure.
It progresses slowly. We now know the root cause of the disease. It's these propagating toxic forms of a protein called amyloid beta. To actually have or see any signs of Alzheimer's disease in a patient, of the 100 billion neurons we have in the adult brain, about 10 billion or so have to be destroyed by these toxic forms before any signs are seen whatsoever. And on average, this takes about 15 to 17 years. Initially, seeds of these toxic oligomers start in the brain region called the hippocampus. This is where short-term memory, recent memories, if you like, are stored. So the first signs of Alzheimer's disease are often related to short-term memory problems, also called cognitive impairment. From the hippocampus, then these destructive forms, toxic forms of amyloid beta progress to the frontal lobes, the front of the brain, if you like, and their behavior, emotion, other difficult problems with Alzheimer's disease start to emerge. But it begins in the hippocampus generally with memory disturbances.
Okay. And, when we think about Alzheimer's, we generally think it affecting the older population. Can you expand on that a little and just say who does it affect? I mean, is there any specific type of individual that's more susceptible?
Well, the answer is yes and no. We are all susceptible, if you like. Age is a clear determinant. The prevalence goes up very significantly with age generally. However, there are individuals who are at a higher risk than the general population. Those with a family history, of course. Those with a certain genetic profile called ApoE4. There are other factors we really don't know. There's one group of subjects called patients with Down syndrome. That's trisomy 21. Who have 3 chromosome 21s, and the gene that codes for amyloid beta sits on chromosome 21. And we often see, in addition to the mental retardation in Down syndrome, often as early as the subject's teen years or early adulthood, a significant and severe Alzheimer's disease syndrome. Full-blown Alzheimer's affects about 5.5 to 6 million individuals in the U.S., but the total population at risk, whose brains are undergoing these terrible destructive changes, might be up to about 40 million people, but it's not symptomatic in all patients early on. So we've got a slowly progressive disease, full-blown Alzheimer's in over 5 million, and up to 40 million who are developing the disease. It just hasn't declared itself yet.
Okay, so definitely a very important disease that needs attention. And
absolutely.
The problem is, though, that I think, so far this year we've seen numerous failures announced, phase 3 advanced trials after a lot of money's been spent. It's been a tough year. What's gone wrong?
Well, it's been more than a tough year. It's a decades-long process, and maybe we can come back to that point. But there have been multiple failures, but there is one ongoing success, a product called aducanumab from Biogen. What we've learned, though, over the last 2 decades or so of clinical trials in patients with various types of products that block amyloid beta, we've learned that only one of the 3 main forms of amyloid beta is toxic. There's a form called monomer, which is like a single strand of amyloid beta. It's not toxic. It's inert. The plaque, amyloid beta plaque, which was widely touted as a cause or driver of the disease, we now know is inert and certainly not toxic. The only form that is toxic are these so-called misfolded oligomers. They're bunches of 2, 4, and 12 strands that stick together, have a tendency to misfold, and when they do this, they become toxic and spread throughout the brain. So products in the past that have all targeted either plaque and or monomer are destined to fail because those aren't the right targets. But that's fairly recent learning where we've really seen that it's this single oligomer-type form, this twisted, misfolded, toxic form that's the real guilty suspect here, and that's what we need to target with our drugs.
Okay, and what specifically gives you confidence that the oligomers are the problem?
There's a massive scientific evidence supporting this. But let me just give you 2 pieces of information to give this some clarity. One, the results of human trials, as I've indicated, that's the best animal model there is. Of course, I don't mean that facetiously, but results in humans really show us what may be working and certainly what not to target, what not to do. So we've learned a lot from the clinical trials, and I don't want to underestimate the importance of that and the hundreds of millions or billions of dollars that have been spent by big pharma. Second, I'll just cite a whole set of experiments that are done in cultured human neurons. So we can grow human neurons from the hippocampus region or other regions of the brain, and then we can test and see which forms of amyloid beta or other, nasty substances may be destroying these nerve cells. And what do we find? The monomer form has no effect on these nerve cells, these human nerve cells that are growing and living. The plaque has no effect on them, but the toxic misfolded oligomers destroy them, often within 6 hours. They're very, very deadly. So targeting the right form of amyloid beta selectively and exclusively is the key to success, in our opinion.
Okay. We only have about a minute and a half left in this segment, but I just wanted to cite a statistic, and then maybe we can chat about it a bit after the break. But according to some of the leading Alzheimer's organizations, estimated costs for not finding a cure are around $200 billion for paid and unpaid care, and then another $236 billion for direct treatment, with much of that picked up by Medicare in the U.S. So maybe after the break, we can just chat for a bit on what stakeholders are doing to address this problem. And, when I think about stakeholders, I mean, it's obviously companies like ProMIS, the FDA, Alzheimer's organizations, and then other government entities. So maybe just a comment or two on that in the last 45 seconds or so, and we can delve into it a little bit deeper after the break.
Yeah, as you pointed out, John, Alzheimer's disease will bankrupt our healthcare systems if it's not effectively addressed in the, over the coming years. Multiple stakeholders are involved, and there are many things that are being done at the level of the regulatory authorities, FDA and EMEA in Europe, Alzheimer's associations, and a very strong push from patient associations and awareness in general. So a lot is going on, not just drug development.
Okay, thank you, Dr. Goldstein. We'll be back in a minute.
All right. John Vandermosten is hosting that segment. We're so happy he's come to us through Zacks Small Cap Research, and you've been listening to Dr. Elliot Goldstein of ProMIS, symbol ARFX in the U.S. and PMN on the Canadian exchanges. This is CEO Money with Michael Yorba. We'll be right back after this break. All right, we're going to go back to John Vandermosten talking to Dr. Elliot Goldstein of ProMIS.
All right, thank you, Michael. Well, we're back with John and Elliot. Where we left off, we were talking about how some of the stakeholders, might make some efforts to make some progress in Alzheimer's, just because of all the hurdles that have been faced over the last several months. And one of the areas that I wanted to explore with Dr. Goldstein was just what the FDA has been doing recently to help make the trial design process a little bit easier than it has been in the past. And Dr. Goldstein, can you share with us what the FDA has done recently that kind of changes things a little bit on the regulatory side?
Sure, I can do that. These are really important changes and, follow the science. I'll talk about changes from both the FDA and the EMEA, the European agency, because their positions are very similar. So past trials treated patients at various stages of disease, mild to moderate, and required two primary endpoints: cognitive status, memory basically, and also functional status, ability to get along in daily life. Now, new guidance from both agencies, and which is very clear to us in the field, is to treat early in the disease and not at the mid or late stage where there's little hope. Early in the disease, when only cognitive impairment is present, i.e., just memory disturbances and related issues, but no functional disability. That leads the regulators, FDA and EMEA, now to recommend the use of only one primary measure of success, i.e., a battery of tests evaluating cognitive function, memory. So this is a big change in many directions. The FDA and EMA, however, are requesting that the companies who are developing drugs in this field also develop so-called biomarkers to give extra support, if you like, to the single outcome now that we're looking for. So all in all, this should speed up trials and make them much more focused and improve possibility of good results.
Yeah, that's an exciting change that hopefully will have a positive result. One last thing that I thought we might cover today is your development platforms that have really distinguished your products. So can you chat for a bit about that? And I think we only have about 1 minute left, but it's a very interesting thing that I think listeners will really be interested in hearing.
Okay, so I'll give you the really short version. A unique and proprietary technology platform consists of two complementary methods. They allow us to predict and identify targets called epitopes that are uniquely displayed on the surface of toxic misfolded oligomers. Normally, these epitopes or areas are not visible, if you like. We identify, I'm sorry that there was a little hiccup on my phone. We identify not only the sequence of the exposed target, but also its shape, called the conformation. We can then make antibodies that are highly selective for these toxic misfolded forms. And there's different antibodies driving different neurodegenerative diseases. We talked about amyloid beta, toxic aggregated forms of alpha-synuclein cause Parkinson's disease.
Dr. Goldstein,
yes.
Please hold on to that thought. We're going to put you on the next segment. We're going to take a break, and we'll be right back with you and John Vandermosten, okay?
Okay, got it. Thank you.
We'll be right back with Dr. Elliot Goldstein of ProMIS and John Vandermosten with Zacks Small Cap Research. You're going to listen to CEO Money with Michael Yorba. We'll be right back. Welcome back to CEO Money. I'm Michael Yorba. We're talking with John Vandermosten of Zacks Small Cap Research. He's brought a guest with him, Dr. Elliot Goldstein of ProMIS. All right, John, pick up where you left off, please.
Certainly. So we were just talking a little bit about the company's or ProMIS's platform, which they've used to develop some very sophisticated drugs, monoclonal antibodies, actually. And so, Dr. Goldstein, maybe, since we have a little bit more time than we thought, can you maybe go into a little bit more depth on that topic, on how you develop these and how it's differentiated from what anyone else has out there?
Sure. Just a quick description of the technology. It's based on two complementary methods that are proprietary that allow us to predict and identify targets, so-called epitopes, that's a target for an antibody, that are uniquely displayed on the surface of toxic misfolded oligomers. And now we know that these toxic misfolded oligomers are drivers of various neurodegenerative diseases. So we talked about amyloid beta at the beginning section in Alzheimer's disease. We've also now identified several new targets on a protein called alpha-synuclein. When it misfolds and aggregates, it's a root cause of Parkinson's disease and a terrible disease called Lewy body dementia. That's what Robin Williams died of. We've also been able to identify targets, epitopes, and create antibodies against toxic forms of a protein called TDP-43. It's a long name, but essentially it's a protein, when it aggregates and is misfolded, becomes toxic and is a known driver of ALS or Lou Gehrig's disease. One of the challenges is in big pharma, our technology, if you like, is the only one we know of that allows to identify as precisely and selectively as one can these targets and then to make antibodies that are selective for them. Usual techniques, so-called physical techniques like X-ray crystallography, which is widely used in big pharma to understand the structure of molecules like these misfolded proteins. Well, you can't use it. It's impossible to use it. These proteins they're shapeshifters. They're floppy. They have unstructured regions. They can't be crystallized, and they have a very sort of short life in the lab as well. So the only means we know of are the proprietary technologies we have, which allow to visualize these sites and create very selective antibodies against these very toxic misfolded proteins. That's what's special about our approach at ProMIS.
Okay, great description there. I wanted to spend a little bit of time talking about your lead candidate, and the name is PMN-310, and then also kind of combine that with what is going on with Biogen's drug aducanumab. In the research that I've read through, there are several characteristics of PMN-310 that mean it could actually be better than the initial indications on aducanumab. Can you compare and contrast a little bit about the two drugs and maybe also go into a little bit more detail about what PMN-310 does?
Sure. Well, let's start with aducanumab from Biogen, which is a very exciting product. Those of us in the field are really excited about this. The first and only product to date to show that it can slow or stop the cognitive decline, the memory disturbances in early Alzheimer's. So aducanumab binds and blocks toxic oligomers, hence its effectiveness, because we know that's the guilty suspect, if you like. However, it does have a downside because it also binds plaque, which we now know is not the target. Binding the plaque is associated with a side effect of brain swelling at the dose that works best with aducanumab, and approximately 30 to 50% of the patients given that dose, which should work, well, they can't take it. They're unable to take an effective dose because of the side effect. That's the main issue with aducanumab. However, in those patients who tolerate it at the dose that is effective, 10 milligrams per kilogram, it's a very exciting product. We believe PMN-310 has great potential as best in class, versus aducanumab. Our product is selective for only the toxic oligomers of amyloid beta, so no off-target waste of product on monomer and plaque the target. So, spending your ammunition on that is just a waste. And we anticipate fewer, if any, side effects, especially related to binding plaque, because we don't bind plaque. So compared to aducanumab, PMN-310 shows virtually no plaque binding, and hence we anticipate being able to give much higher doses with little or no risk of brain swelling. Higher doses, greater efficacy. That's what we expect.
Okay. Well, actually, what I hear you saying is that it's more targeted and there's less risk to it, I guess, in summary.
Well said, John.
I want to get an idea of just the timeline on PMN-310. Right now, you're doing preclinical work and preparing to go into the clinic. Can you give us an idea of what the timeline might look like? When will you have first human trials, and when might it be approved by the FDA if they so choose to do so?
Okay, that's quite a long road, obviously, or road to hoe, if you like. First step, we announced last week that we initiated manufacturing of PMN-310, which is sort of the first step onto, submitting an IND or that's your regulatory request to do clinical trials. So the steps that are left to complete now is to do all the manufacturing, scale up, characterize the product, etc., over the coming year, and to do preclinical ecology studies in mice or rats and a small primate. That's absolutely required and necessary. We announced that we anticipated entering phase 1, the first clinical trials, in the second half of next year. I can't be more precise on the date, only because we're just starting now to line up meetings with the regulatory authorities in the United States, Canada, and England, actually, and perhaps in other countries as well. So once we have more clear feedback from the regulators, have an agreed program and plan, the timelines will sort of sift out of that, if you like. I think important point from the timelines, we expect to be in the clinic the second half of 2019, and aducanumab, which is currently in phase 3, the market anticipates that the results will be available in early 2020. So we anticipate, launching our trials in 2019. So we should have some of our first clinical data at about the time the final data from aducanumab emerges, and our anticipation is that data from aducanumab will be positive and the field will become very exciting again. So from that standpoint, the timing is very important. In terms of commercialization, it's a little too early to look out to that right now. Let's get into humans first.
Dr. Goldstein, it's been a pleasure having you on the show. We've got to close for now, but I want to thank you for being a guest on our show.
Thank you for your time. Appreciate it.
My pleasure. You've been listening to Dr. Elliot Goldstein of ProMIS with our in-host correspondent, I should say our biomedical and biotechnical correspondent, John Vandermosten from Zacks Small Cap Research.